everyone. In this video, I will discuss about the morphology of viruses. Please like this video and subscribe it. I am Naina. I will start now. Here, I will cover some topics like viruses, definition of viruses and structure of viruses, shape of viruses and sizes of viruses. Finally, I conclude that transmission of viruses. Now, I will start with definition of viruses. What is meant by virus? See here, virus is the Latin word. It means poison. Poison means it is very dangerous. Okay. This virus first discovered by Ivanovsky in 1899. He is a scientist. Ivanovsky first discovered virus in 1899. Viruses are the smallest known infective agents and perhaps simplest form of life. See here, viruses are very minute structures. These are infective agents. Why? Because it is easily spread one animal to the another animal diseases. Okay, disease easily spread through the one animal to another by help of the viruses. Its lifespan also very smaller, like 1 hour or 24 hours, maximum 24 hours. Next we go to the morphology of viruses. Morphology means some characters. Okay, like viruses are extremely smaller in size. They are smaller than bacteria. These are the very minute particles, like very smaller in size, and compared to bacteria, these are the smaller. Okay. Next, size range. How much size be seen here? 20 to 300 nanometer. We measure minute particles in nanometers. So, this size 20 to 300 nanometer. They cannot see under light microscope, hence we call as the ultra microscopic. Like these are very minute particles, we already know about that. We can't see by use of the light microscope, so we call as the ultra microscope. When we use advanced method like electronic microscope, that time easily we identify. Okay. Next, they do not have cellular organization. Why? Because these are minute particles. We can't see any cellular organization. Like for example, digestive system, reproduction, and like reproduction we see, but some sexual and asexual binary reproductions, these type of the reproductions we see. Okay, but completely we can't see the cellular organization. Why? Because these are minute particles. Next, they contain only one type of nucleic acid, either DNA or RNA, but never both. Here, nucleic acid, how our cell also have DNA or chromosomes, that like virus also have nucleic acid in center part. It may be DNA or RNA, but we can't see both in one virus. Okay. Next, they are obligate intracellular parasite. These are the obligate and intracellular parasite. We see in the innermost layer of the our host body or host body. Next, extracellular infections viral particle is called virion. The viruses affect in external part like skin. For example, we given skin. In skin, these small particles like virus particles infected to the our skin, that particle we call as the virion. Next slide, shapes of the viruses. The various shapes of the viruses we seen here, like for example maybe rod shape, bullet shape and spherical, oval, irregular shape, rod shape and brick shape or complex shape. This type of the shapes we seen in the viruses. For example, I given here fox virus. This is a fox virus, are brick like shape. We already know about that brick, where you use like construction of the house. Okay, that like we seen in the fox virus. And rabies virus, these are the bullet like shape. Okay, next tobacco mosaic virus, these are the rod shape. I will see next slide here. Shapes, this is the clear picture for you all. This is the round shape virus, thread like, rod like and bullet like. Next robot virus, like we call as the bacteriophage, like complex. Next, these are the polyhedral and helical form is there, spherical form is there. Next, complex. Complex also we call as the bacteriophage. Next we go to structure of viruses. What is the structure of virus? See, this is the structure of virus. Here outermost covering layer we call as the envelope. Okay, this is envelope. And the innermost layer, double layer, that we call as the capsid. The capsid made up of the protein coat. And in this capsid, small subunits we seen here, like triangle like, that subunits we call as the capsomers. And in envelope, outermost envelope, on the envelope some projections like pin like structures we seen, that we call as the spines. Okay, and innermost layer, that particle present, that we call as the nucleic acid. Okay, now see here, capsid. Capsid surround the center of the virion. See here, second layer of the virus, that we call as the capsid. It is center part. Next, nucleocapsid. It is combination between nucleic acid and the capsid. The combination between nucleic acid and capsid, that we call as the nucleocapsid. And next one, membrane or envelope. This surrounds the nucleocapsid. The nucleocapsid surrounded by the layer, outermost layer, that we call as the membrane. And spikes. Spikes nothing but these are the protein spikes. It is helpful for the to attach themselves to the host body. It is helpful for the viruses to attach themselves to the host body. Next slide. Viral capsid. Viruses consist of nucleic acid core surrounded by the protein, that we call as the capsid. We already know about that capsid definition. The nucleic core surrounded by a layer, that is protein coat, that we call as the capsid. Next, capsid is composed of large number of capsomer, which is made up of the polypeptide molecules. The capsid contain large number of the capsomers. These are how polypeptide molecules. Okay. Next, capsid with enclosed nucleic acid is known as nucleocapsid. We already know about that. The combination of the nucleic acid and capsid, that we call as the nucleocapsid. Okay. Next, functions of the capsid. What are the functions? See here, it protects the viral genome. Here, viral genome nothing but DNA or RNA. It will be protect like from physical destruction or physical damages. Okay. Next, it serves as a vehicle of transmission from host to another. We commonly call as vehicle of transmission. Why? Because it easily to transport diseases one organism to the another organism. Okay. Next, it provides the structural symmetry to the virus particle, like it is helpful for the to give structure or shape to the virus part or virus like body. Next, viral symmetry. Next we go to viral symmetry. Here we discuss about the architecture of virus. Here we consider as we divide three groups based on the arrangement of the morphological subunit. Some particles, how it will be arranged, that we call as the morphological subunits. Here see, there are three types we already know about there. Following there here, isocahedral symmetry, helical symmetry and complex symmetry. First one, we start with isocahedral symmetry. This is the structure of the isocahedral symmetry. And isocahedral in this word will be split. Isoca means, it is a Greek word, it means 20. It means 20. It is a polygon with 12 vertices or corners. See here, you count this picture, 12 vertices or corners we see and 20 sides. 20 sides are there, this like, and each side is in equatorial shape, like triangle shape, equatorial triangle shape. Next, pentagonal capsomers at the vertices, that we call as the pentagons. Pentagon, you know, like this like which thing, that we call as the pentagons. And hexagonal means here 6 sides we see. Okay, hexagonal capsomers making up these sides, that we call as the hexagons or hexons. Here, for example, we given adenovirus. Okay, this is the structure of the adenovirus. Next, we go to helical symmetry. The nucleic acid and capsomers are wound together in the form of helix or spiracle. Here, nucleic acid and capsomers are wound together or clamped together to form the helical or spherical shape, that we call as the helical symmetry. For example, we given here helical symmetry, influenza virus, like nothing but common cold, okay, and rabies virus and tobacco mosaic virus. These are the example for the helical symmetry. Next, complex symmetry. Viruses which do not show either helical or isocahedral symmetry due to complexity of their structure are referred to have complex symmetry. Here we can't see like isocahedral or helical. It will be differ to these shapes. It is very complex structure, that we call as the complex symmetry. For example, Fox virus. It is also called as the bacteriophage. We also seen in the bacteriophage. Next, viral envelope. Here we divide into two types, like maybe enveloped and non-enveloped viruses. First, we discuss about the enveloped viruses. Outer covering of virus containing lipid is derived from the plasma membrane of this host cell during the release by budding from the cell surface. See here, envelope means outermost covering layer will be seen, that is made up of the lipid. It is from plasma membrane for the host cell. Okay, and it will be released by the budding process on the cell surface. Next, the envelope is glycoprotein in nature. It contains glycoproteins in nature. And for example, we given here hepatitis B, HIV virus and herpes virus. These are the example for the enveloped viruses. Next, non-enveloped virus. Viruses which does not have outer covering, like compared to envelope, here we can't see outermost layer. Okay, so this virus we call as the non-enveloped virus. Here see diagrammatic representation, envelope and non-envelope is there. Here examples also I given. Next, the outermost layer, this layer we call as the envelope. This is made up of the proteins and lipids from the plasma membrane. Okay, next, non-enveloped. Here we can't see outermost layer. Why? Because like non-enveloped we call as the non-enveloped. In innermost we see the nucleic acid, that may be DNA or RNA. Here example for the non-enveloped viruses: norovirus, parvovirus and HAV and HEV. These are the example for the non-enveloped viruses. Next slide. Peplomers. This is the structure of the virion or virus. Okay, here papillomas means immature virus particle contain glycoproteins, and here we see some projections like pin-like structures. We call as the spikes on the outer surface. This process we call as the this envelope we call as the papillomas. Papillomas means immature virus particles. The glycoproteins often appear as projecting spikes on the outer surface of the envelope, which we call as the papillomas. A virus may have more than one type of viruses. Here we see more type of the viruses. Okay, it will be differ. Now, functions of papillomas. It helps for attachment of virus to host cell receptors to initiate enters of virion into the cell. See here, papillomas are very helpful for the viruses. Why? Because to attachment or to attach the like host body cell, and it is helpful for the to enter the host cell. Okay, and next one, it attaches to the receptors on red blood cells, causing these cells to agglutinate. Like here, when it will be attached to the blood cell or RBC cells, it will be causing agglutinate, like clump the cells. And next one, it has antigenic or antigenic property. It acts as the antigenic property. Next we go to viral nucleic acid. Here we see nucleic acid like genetic material, we call as the genetic material DNA or RNA. Okay, viruses contain a single kind of nucleic acid, either DNA or RNA. Here we see RNA genome. Okay, sometimes some viruses have DNA, which encodes the genetic information necessary for replication of the viruses. It is helpful for the genetic information to give the genetic information and replication process. Next, genome may be single strand or double strand or non-segmented. Here, genome like DNA or RNA may be single strand or double strand, or it may be segmented or non-segmented we see. Next, according to nucleic acid present, viruses can be classified into two types. These are DNA virus and RNA virus. Next, transmission of viruses. As with many infections, viruses can be transmitted between susceptible individuals by a variety of means. See here, virus transmission may be dangerous, like it may be affected by water. We commonly see water, air and food. Okay, next, the details provides related mainly to viruses infecting humans. Many animal viruses do not remain infectious for very long outside of the host. See, we can't see like transmission of the animal viruses on the humans. Why? Because these are not affected severely. Okay, next, respiratory. For example, we given here influenza virus, we called as the common cold. See here, a person to suffering from the common cold, some symptoms we identify. These are sneezing and coughing. When he cough, in that time he releases more number of the viruses in the air. When we inhale that air, easily virus will be attacked to our body, enter to the our body. Okay, because of very sensitive viruses easily to enter our body, and also when we contact closely to that affected man, easily we suffering from the influenza A virus. Next, sexual transmission. For example, we given here HIV. A person suffering from the HIV, when we contact with sexually, that time easily it will be transfer. Okay, again when we use a person, HIV patient will be use some needles, again sharing with the another healthy persons, that time also easily the HIV will be transfer. Next example for the rabies, we seen in the animals. For example, dog, when we already know about that, like dogs saliva contain the some viruses, that is major rabies. In many instances, the virus infection is a specific pathogen of the animal and is not normally transmitted to the humans by any other mean. See here, rabies, it is transmitted to the only in animals only, but not transmission or affected to the humans. Okay. Next, blood-borne disease. For example, hepatitis B. Transfer through contaminated blood products or via shared needles with drug abuse. A person to habit or to adapt to the drug injuries, that needles again we share to the another person, healthy person, easily blood-borne disease will be transferred. And contaminated blood products means blood and like semens and vaginal fluid. Okay, these are the blood products. See here, blood-borne viruses, we commonly short form BBV. These includes hepatitis B, hepatitis C and HIV or AIDS. Okay, how it will be transferred, you see, transmitted via contamination by blood or certain body fluids. For example, blood, vaginal fluids and semens. These are the fluid-like structure, fluid forms. Next, let us we summarize what we have learnt in today's session. Okay, one by one we see here. In Latin, virus means poison. Okay, it is poison means dangerous. Next, the first virus was discovered by Ivanovsky. He is a scientist. He discovered in 1899. Now, definition of viruses. Virus is a smallest infectious particle, unicellular, obligate, intracellular parasite. Okay, these are the smallest particles, like maybe in unicellular and it is intracellular parasite also. Okay, let us see some characters here, important characters. We discuss about characters like nothing but, first one we discuss sizes. Sizes 22 300 nanometer. Shapes like bullet shape, brick shape, complex, helical, these are the shapes. And structure of the viruses, major parts, capsid, envelope, nucleic acid. These are the structures. See, let us see here. Next slide. Size range 22 300 nanometer. Shapes may be rolled, bullet, spherical, oval, irregular, bricks, etc. Okay. Next, they do not have cellular organization. Why? Because these are minute particles. Okay, we can't see any cellular organization. Next, they contain maybe DNA or RNA, like nucleic acid, DNA may be RNA, and but we can't see in particular a single virus contain both nucleic acid. Okay, it will be absent. Next, intracellular infections, viral particle we call as the virion, which particle will be infected to the viral, that we call as the virion. The structure of the virus is capsid, nucleocapsid, and membrane or envelope, protein spikes. Spikes nothing but pin-like structures we see in the outermost layer of the virus. Viruses consist of nucleic acid core surrounded by protein is called capsid. The nucleic acid core surrounded by the proteins that we call as the capsid. Next, capsid with enclosed nucleic acid is known as the nucleocapsid. Why? Because these are the combination of the capsid and nucleic acid, so we call as the nucleocapsid. And in mature virus, the glycoproteins often appear as projecting like small projection spikes seen on the outermost surface that we call as the peplomers. And viral architecture can be grouped into three types based on the arrangement of morphological subunits. They are as follow now: icosahedral symmetry, for example adenovirus, and second one helical symmetry, for example rabies virus, influenza virus, nothing but common cold virus, and third one complex symmetry, example fox virus. Next, virions may be enveloped or and non-enveloped. Two types we divide here. Enveloped, example HIV, hepatitis B, okay, or etc. And non-enveloped means it not contain outermost layer of the virus like lipid layer, for example norovirus. Next, transmission of viruses by may be respiratory, like when inhale the air, that time easily a virus will be entered to our body or host body. For example common cold, like we call as the influenza virus. And next one, sexual transmission, we already know about that, HIV. Okay. Next, blood-borne diseases like hepatitis B. These all are summarization I given here. Any doubt means you ask in comment box. Okay. I hope to understand or to understood this concept and like this video. Thank you. Please support me.